So really, I'm going to talk to you today about 3D printing and its applications for a whole range of science, not just making things, but how do we actually make ourselves more creative as a whole? So the talk title is 3D printing for speeding up scientific development, from personalized drug delivery to continuous flow synthesis. And you'll see as I go through the talk how this has actually impacted a lot of what we do based on the ability to do things because it has given us the freedom to carry out things in different ways. So in terms of our laboratory, we really think about it as a combined laboratory. So where we carry out our chemical synthesis, we've now integrated 3D printing into that. We've also got low-cost electronics and AI, and more recently, virtual reality. But what we've actually found is that the laboratory's become a combined laboratory. We effectively think of it as a cloud-based element through all these different factors. In terms of my research, we do a lot of medicinal chemistry, looking at anti-cancer drug discovery, neuroscience drug discovery. And in terms of making these molecules, we often encounter challenges in the ability to actually manufacture these or synthesize these. And so one of the things we turned towards was a lot of the time was 3D printing, because it gave us the ability to think about the way we can do catalysis. So one of our early bodies of work focused on the embedding of catalysts into stirrer beads. And you can see the example on the top right shown here. This allowed us to actually make molecules simpler and easier and reduce the purification steps required in that process. When we started to move on to the scale-up synthesis of some of these compounds, we found actually the technology wasn't there to enable us to do this. And we started to use 3D printing to actually manufacture our own flow systems, enabling us to carry out synthesis on a large scale. As we went into the pandemic and working more collaboratively, we found there were difficulties of meeting up people around the world. And we moved into virtual reality in that space and low-cost robotics, which I won't really talk about today, giving us the ability to actually work together across the globe. As well as synthesizing molecules in small and large scales, we've also started to think about how do we actually get the right dose for the right patient at the right time. And with 3D printing, you can do this, and we'll talk about this a bit in the talk today. The ability to create tailored dose systems, shown on the bottom left, to give a steady state release for a patient, or for more personalized delivery based on the patient's needs, with GRAS, or generally regarded as safe polymers, giving us that flexibility. Now, in terms of my laboratory, a lot of our work has gone into actually making this work efficiently, be it the ability to work at the fume hoods, where you can actually carry out the chemical synthesis, working in the laboratory, at the back of the laboratory on the analytical systems, or when you're working at the bench with electronic lab notebooks, and then translation of that into the office. It's that seamless ability to navigate data in the lab through to the office as and when you need it. So why 3D printing? Why is it so important? Well, the work that we do focuses a lot on SLA chemistry, or SLA 3D printing, or FDM printing, fused deposition modeling. And it's the ability to manipulate materials depending on your requirements, what you need, to make different structures depending on what you want, and then to apply those to a range of applications. So 3D printing gives you the control of all these three different factors. How does it work? Well, with FDM printing, you take a thermopolymer, pass it through a hot nozzle, melt it, lay it down to a structure shape, and this gives you a three-dimensional object shown below over time. So the motor goes back and forward, creating the object that you need for the application that you want. SLA printing is slightly different. Here we look at a range of monomers, or liquid monomers in this case, which are in a tray. A light then polymerizes those monomers to polymerize those into a structure, and then creates the next layer of an object, as you can see below, to create a three-dimensional object that you require for whatever purpose. And so these are the two main types of 3D printing that we use in my lab for different reasons. However, they are somewhat expensive. So the printers themselves are roughly about £2,500 each, or the Ultimakers around about £4,000, depending on what you're doing. So we have spent a fair bit of money on this, roughly about £40,000 just on 3D printing for equipment and other applications in the laboratory. Our virtual reality costs are also quite high for collaborative work, so around about £15,000. So our total investment in this area is roughly about £50,000 so far. So it does seem quite expensive. So why do it? Well, this is what it's saved us in the laboratory alone. Over the last 5 years or so, we've saved approximately 1.8 million on equipment and different things in the laboratory just by having the ability to 3D print. So that investment has realized there's huge savings just in my laboratory alone. Examples of this can be seen quite quickly in the next slide. So if you go into most academic laboratories and probably a lot of industrial ones, you will see these horrors in the fridges and freezers. You have 10 or 20 years of research balanced on a glass shelf because of the lack of these flask holders because they're expensive. Alternative people use bottle caps, depending on what they're doing. But that's a crazy way of doing things. You have upwards of 10 million pounds worth of research balanced on an old bottle cap or an old cork ring, simply because the commercial ones, shown in the bottom right, the red ones here, are expensive. Why not think about 3D printing to solve this problem? And here you can see our solution above. These now are very, very low cost. At 50 to 70 pence per unit, this is a huge saving on the currently £23 per unit of the plastic holders. So you can easily see here how we can save a huge amount of money just on applying 3D printing to these one thing. And that £3,000 cost is the cost of a printer. So you can easily see now how 3D printing can help you save a huge amount of money and vastly speed up your scientific research. If we were to implement this at UCL in 2015, we thought this is what we would save. 25 million. This was a really conservative estimate a few years ago. And at the time, we estimated about £10,000 saving per laboratory just by doing the equipment, which we thought was a lot at the time. But you can easily see how, with UCL being quite a big university, you can save a huge amount of money quickly and easily just by implementing 3D printing. Now, that was savings from about 10 years ago. What we've now learned is that rather than that £10,000 saving per lab, we can easily save between £500,000 to £2.5 million, depending on the applications that you do. So 3D printing has huge applications for speeding up the science that we do and making it far more accessible. It also works on other cases. Here you can see some very happy PhD students in the lab, where being in a London space, it's difficult working conditions. The space isn't fantastic. You can see here there's lots of stuff on the benches, the keyboards are always spread out. But actually with 3D printing, you can solve a lot of these issues. You can create holders for the keyboards and holders for the mice, bringing up to the bench space. And the nice thing with this is you can share that design with anyone else in the world. And again, if you look at my Thingiverse account, you can find this and other examples for whatever you want to modify and change depending on your local requirements. In terms of 3D printing and its approach for personalization in medicine, there are huge opportunities with this. It's not just medicine, but also devices. Because again, we can personalize drugs, we can 3D print these for ourselves, but we can also develop personalized devices for patients as well, with personalized dosing. We can also think about applications externally to patients, because again, we can scan them, we can develop personalized wound dressings, and we can then develop personalized approaches that match a patient's physiology and body orientation or shape or drug delivery for internal cavities of patients. In this approach, published in 2017, we looked at the personalization of dosing for patients for wound healing, the ability to incorporate antibacterial agents into 3D printed plastics, 3D print those to match a patient, gave us the huge ability to control drug delivery for patients. If you think about applications for patients externally, you can easily think about situations where patients have broken bones. And again, you can generate a personalized cast for those patients by taking the unbroken arm, mirroring that, and generating a personalized cast for those patients. So again, just think about applications. It's not just about medicine. It's the applications of patients for personalized devices, depending on what patient needs. We can easily think about printing within patients, because again, with SLA-based resins, we can actually take personalized doses and place those within patients with laser-like precision, placing the dose of drug exactly where we need it. So again, if you think about tumors, for example, for a non-surgically resectable tumor, we can easily place a dose of drug right next to those patients and where they need it in the delivery space in the patient's body. So you can see here how we can do that. With 3D printed pen, we can easily then extrude a polymer containing a drug and actually place that in the body cavity depending on what we need as a surgical adjunct. We can also think about personalized doses for patients as well. Because again, when we take a tablet, there is the issue of the tablet breaking down, passing through the stomach very quickly and easily. But we can also think about slowing down drug release. In this way, we can incorporate drugs into patients by a tablet that you swallow. This then gets bigger in the stomach and slows down the release in the stomach into the intestines, enabling sustained drug delivery for patients. In this manner, we avoid the up and down one-day type release of tablets, giving a nice steady state release for patients over a 10-15 day period. How does it work? Well, when you swallow a tablet, the gelatin capsule breaks down, the drug device grows in size, and then releases dose in the patients over time, allowing for nice steady state release depending as required. We can also modify the printers as well with 3D printing because we can modify the build plate to take into account balances to make sure that a dose for the patient is accurate and reproducible, making a nice steady state way for drug delivery for patients. Now, if we move on to 3D printing, it's not just about the fact that we can actually make things, it's also about the CAD design. The ability to actually interact with the objects within virtual reality and then speak to AIs of patients, enabling us to collaborate with researchers from all around the world, speeding up development as a whole. But one of the huge areas where 3D printing has implications are in the way it can change the way we do things. So going back in time to when I graduated in 1996 from my undergraduate degree, you see that laboratories haven't actually changed that much. So roughly about 10 years later, we were still doing things in exactly the same way in laboratories. We're just starting to see the entry of electronic lab notebooks into laboratories. But in 2021, just a short time after, you can see how laboratories have advanced enormously. We have the ability now to actually 3D print holders for small tablet PCs that can then be added to fume hoods, enabling us to have digital connectivity in fume hoods, which traditionally has not been possible at all. This vastly speeds up the way we can do things, enabling us to work far better together in buildings and in laboratories together just by having the adaptation and thinking about how we can modify and improve things wherever we are. So that change in our approach to chemistry can also be applied to the way we carry out chemical synthesis. Because in 28 years, going back to 1996 to now, chemistry has mostly stayed the same. The way we carry out reactions has pretty much stayed the same, where we weigh out reagents, add them to flasks. We carry out reactions in the same way with an internal magnetic follower. We clean up the reactions in pretty much the same way: filtration, separation, purification. And purification has improved, but we still use column chromatography in a lot of cases. And a lot of these problems arise because the inefficiency of reactions. Our advancements really have also been helped by technology, the ability to speed up reaction setups with multi-well reactions, the ability to use robotics for microwave-based reactions, and cleanup of reactions via purification via biotage or automatic purification rather than the column chromatography shown above, all speed up chemical synthesis. But what we wanted to think about was a new way of carrying out chemical synthesis. The ability to change the magnetic follower in the reaction flask to embed a catalyst into the external structure allows us then to carry out chemical synthesis in an entirely different way because it speeds up this entire process. The reaction setup is faster because the catalyst is already embedded in the device. The reactions are also faster because you're not waiting for mixing to occur. The cleanup is simpler because you take your catalyst out at the end of the reaction. And the purification is also faster because there's less byproducts in the reaction. So how does this work? Well, when we think about adding catalysts to reactions, most of the time we add these externally dropwise to the reaction flask. The problem with this is that the reagents are added to the flask don't always mix efficiently at the same time. If you look at the central reaction column in this case, the reagents are mixing in a poor fashion because then an inefficient ratio in the center of the reaction column. Our reagents that are mixed in the system with a catalyst don't always mix in the same way. So the way we thought about using 3D printing to change this approach and change the the way we do chemistry was to take our magnetic followers from a flask and modify them so that they contain an external catalyst around them, allowing us to mix and catalyze reactions at the same time. In this way, it's easy to add to reactions. There's no weighing out a catalyst needed, and we can simply remove the catalyst at the end of the reaction, resulting in cleaner reactions. So the idea behind this approach was the fact that our catalytic devices help chemists reduce costs and time taken to discover new medicines. The idea behind this is we have simple color coding. So you just take your catalyst out of the espresso pod, add it to a reaction, and then you're good to go. The nice thing with these is that we can actually develop these with 3D printing. Here we have SLA-based printing, so the liquid polymer shown before. We had a catalyst that liquid monomer, polymerized that to create this solid structure shown here on the bottom left. We placed that simple stirrer bead in the cavity, shown here on the left. And this now allows for far faster mixing of reactions and makes this procedure far simpler and easier to do. And in this manner, we can easily redesign these whole range of reactions just by modifying the design that we want to match the reaction that we want to do. Mixing effects is enormous with this process because from a simple stirrer bead, which just simply mixes the reaction shown here in these sort of computational methods, the mixing from our 3D printed devices is far faster, meaning that reagents are mixed in a much simpler manner and flow across the surface in an easier way, allowing the reaction to be catalyzed in an efficient and simple manner. This process works really well. But one of the problems with this is that actually the reagents, when they carry out the reaction, aren't always converted at exactly the same speed. You rely on your two reagents meeting on the stirrer bead, the catalysis taking place to get the product. But what this means is that sometimes the reagents stay in the flask for quite a long period of time, and this can lead to byproducts. So we wanted to look at 3D printing and how we can actually modify this process to give more access to continuous flow chemistry, enabling the reaction to be simplified and made more efficient. Continuous flow chemistry, for those that don't know how it works, focuses on scale up over time. Rather than adding reagents in a bigger and bigger flask for scale up, this focuses on taking reagents that are separate, so reagent A and reagent B. We pass them through a very small tube in packets, in effect. These come together and meet, go into the reactor, get converted through its product, which then passes through in a continuous process, enabling for scale up over time. So only very small amounts of reagents meet at any one time in the reaction flask, making the reaction far more efficient as a whole. One of the problems for continuous flow chemistry is that it is expensive. The cost per system is enormous, around about £50 to £100,000 per system. But it also means when you carry out your work in the fume hoods, they block access to space. We wanted to use 3D printing to effectively redesign these to make it far more efficient for carrying out reactions and also make these far more low cost. Because again, we want to take advantage of the flow chemistry to speed up our drug discovery process, giving us faster mixing, increased safety, and easy scale up. We also decided to work with a commercial manufacturer to develop this and bring this to market. And using 3D printing, we're able to bring this to market in less than 2 years as the ICA Flow. This started with our prototype in 2018, where we 3D printed a system for continuous flow. We developed an early optimized product in 2019. We then met with ICA very shortly after. And by May 2020, we had our first working prototype in the laboratory, and finally the commercial launch in 2020, just by having the ability to 3D print our entire system from start to finish. Some of you may notice that this timeline wasn't a fantastic time to be developing anything, because this was our working laboratory at the time in our garage, where we were using flow chemistry at home and developing products as we could in a fairly challenging working condition. But 3D printing enabled us to do this because we were able to 3D print our prototypes at home, work with others in virtual reality at the time, and collaborate across the globe to bring this to market in a really, really short space of time. Our system is simple because it uses commercially available stirrer hot plates. We can then reduce the need for motors in our system by applying these with compressed air, which passes the liquid through the system into the reactors and then out the other side. And all this system, as you can see, uses CAD design to develop the system, to prototype it, 3D print the system, and actually make it and bring it to market in a really, really short space of time. The other beauties about the system is it uses these very low-cost 3D printed reactors. These are typically about 50p to £1 per reactor, enabling us to make as many of these as we need. Now, one of the beauties about these reactors is that they allow us to modify the flow path, but also to think about carrying out reactions because we want to investigate the science, not to think about the cost of these before we do a chemical reaction. In terms of the development of the flow, we won't go too much into this, but that ability to take our CAD design and then work with others using virtual reality was a key enabler for its development. The ability to work with colleagues in the US, Germany during the pandemic was a key driver in its success and bringing it to market. Because again, those challenges in the pandemic were a key driver of how we brought this to market in a short space of time. Because again, when we were working during the pandemic, we couldn't meet up. Using VR, we were able to do this quite quickly and easily. VR is also a huge enabler for the next steps of the ICA flow. Because again, we can take real-time data and actually interact with others, blending the real and the virtual at the same time with a VR headset to look at reactions from across the globe all together at the same time and work together simply and easily with real-time input of data. And we can do this via the approach ether, which I won't talk about too much to avoid taking up time. But that CAD design can then be extended into virtual reality, where we can now incorporate those designs, telemetry from those reactions, and incorporate them into AI avatars, which can help us understand how the reactions are going. So that digitization pathway of 3D printing has been hugely transformative in speeding up the development from chemical reactions through to digital data and reaction telemetry from across the globe. We have the ability to take those digital information and incorporate this into an AI who can then speak to us about those reactions and help us optimize those reaction processes. So overall, we've been hugely transformative in terms of our group developments, going from flow chemistry to the signature pumps, collectors, catalytic stirrers into VR and AI software, but also ultimately demonstrating that we can actually incorporate all of these elements into our laboratories. And finally, I'd just like to thank everybody for getting us to this stage, and thank you for listening as well. I'm happy to answer any questions.
Hello.
Fantastic. Thank you, Stephen. Do we have any questions at all for Stephen? Oh, there we go. Got something in the chat. Sorry, I thought of quite a few questions throughout the talk. How do you estimate the cost saving of VR training? I'll throw that one to you first, perhaps, then I'll let you know the rest.
So the cost savings of VR training, I guess. So I met a colleague in Thailand last week who wanted training on the ICA flow and how to use it. Now, if they'd have come to my laboratory in London, that would have cost probably about £6,000 to £8,000 in total by the time you take into account flights, travel time, lost time, in meeting up, hotel costs. And in total, they probably would have lost about 4 or 5 days productive time of working in Thailand. Now, they bought a headset at £250. We then met up in the software for about an hour. So from those 4 days, you're down to about an hour in VR, which is a huge amount of saving. So they probably saved about £6,500 just by buying one headset and meeting me in VR. As a rough estimate.
It's a good example.
I would love to have gone to Thailand, but
yeah, that's the only problem, isn't it? It prevents you.
Yeah.
And then what was the next question? So Casey said, how do you validate drug stability within the material you're using to 3D print?
So there are challenges with 3D printing because, again, you are going through a thermal process which does cause drug instability and can cause breakdown. So quite often we'll look at drug release and we'll quantify the drug concentration afterwards using HPLC and check for any byproducts within that process.
And would that modified oral dosing work for drugs that have a higher pH non-polar?
Yes, it can do. You might have to modify that somewhat to make different formulations within the 3D printed material. But again, it's that ability to have capsules within capsules for drug release, which can be quite useful.
Perfect. And then is the amount of catalyst used from the stirrer the same in each reaction, or would it decrease with every use? Can the reactors be reused, recycled?
We can use this about 4 or 5 times without any loss of activity, depending on the catalyst. Some do get degraded. It does depend on the reaction. But it's that flexibility and standardization of chemical reactions across different flasks, which actually make a big difference to the reactions themselves. Does the iFlow have any modular capability with respect to the reactor you can use? Yes, you can do. We do a lot of photochemistry. I didn't really talk about that today to avoid running over time. But there's loads of stuff online you can see about how we use that. It's electrochemistry, photochemistry, scale-up synthesis. It's all possible with that same system. But the reactors are pens because you can make those and 3D print them yourselves.
Perfect. Any other questions at all for Stephen?
Don't worry, Casey, no problem.
Any other questions at all?
Hi, Michelle. Lovely to see you again.
Hi, I've got a question for Steve.
Go
Sorry.
All right.
I could have asked you this next week, really, but actually, you were talking about you were formulating 3D printed treatments for wound care, and I was just wondering about
Yes.
your efficiency of drug encapsulation within that process.
So normally you want to go, well, it depends on the application. So with the photopolymers, you can go up to about 40% loading because it's easy to apply that to a patient's skin. If you want to 3D print something with FDM printing, that tends to be about 10 to 15 to 20% maximum. Otherwise, you get problems with the polymer structure and the shapes that you're creating.
Yeah, I was going to ask about that. And then what polymers are you using for that?
We tend to actually make our lives a bit easier now. So quite often now we 3D print the object and do a post-loading protocol for the drugs because that is a much easier way of doing it. If you are incorporating drugs into the polymers, you need to go through a lot of optimization before you 3D print those. Now we take GRAS polymers and do a post-loading protocol for incorporating drugs into those. It's a lot simpler and easier to do that. And you already have the shape available for the patient.